Hello, welcome to Dolly and the Sciencey Humans. We are your hosts,
Didi,
Emily, and Zeena.
Today we will be teaching you all about cloning.
Clones are two organisms that are identical genetic copies, meaning that they are genetically identical to one another.
Clones can be made naturally, like twins for example, but can also be mixed in a lab in two different ways, the first being artificial embryo twinning or by a somatic cell nuclear transfer.
Let's start off by talking about the differences of each process. Artificial embryo twinning is made to mimic the natural process of how twins are created and developed. When using the embryo twinning technique, scientists will take a very early embryo and then split into individual cells where they are monitored for development on a petri dish. The embryos are then placed in a surrogate mother and carried until birth.
A somatic cell nuclear transfer, also known as SCNT, is when the nucleus is removed from any healthy egg, which then becomes the recipient of another nucleus that is being transplanted from another cell. This can then be used to create an embryonic stem cell, or can be implanted in a surrogate mother to create a cloned individual.
Okay, theory is theory, but have there ever been any successful clones in real life?
Yeah, I mean, for all we know, there could be a secret clone living in the basement of a house somewhere out there.
Well, actually, there have been a couple of major breakthroughs these past 50 years, but the process and gradual discovery of cloning started in 1885 when Hans Adolf Edward Driesch cloned a two-celled sea urchin.
He discovered that by shaking the embryo of the sea urchin, it was possible to separate the cells, which would grow into an identical sea urchin.
This experiment was important because he discovered that each cell in the early embryo had a complete set of DNA and could potentially grow into a full-size organism.
This experiment has inspired many other successes, such as Dolly the sheep in 1996, the first mammal to have been created by a somatic cell transfer.
A somatic cell is any cell taken from a living organism other than a reproductive cell.
Dolly was one out of 277 unsuccessful fusion attempts and was created from a 6-year-old sheep. Ian Wilmot and Keith Campbell created Dolly from transferring the nucleus of an adult sheep's udder cell into an unnucleated cell egg.
We know that every cell's nucleus contains a complete set of DNA, but when we take a differentiated adult cell that has specialized its functions, it can create a problem.
When an adult cell nucleus is used as a donor, we would need to reset its genetic information to an embryonic state. This way, there won't be any functions that are shut down in the differentiated adult cells.
This is the main reason that all of the attempts to clone the sheep had failed, because the resetting process of the cell's information is incomplete and the embryos fail to develop.
The most recent breakthrough was in 2013. Shukrat Metalipov and his colleagues were the first to be successful creating a human embryo from a somatic cell transfer.
Fun fact, in 2004 through 2005, there was a cloning controversy in which South Korean scientists had falsely claimed that they had created a somatic cell nuclear transfer to create embryonic stem cell lines. After this, the scientific community demanded stronger evidence for future procedures if they had actually been successful.
There are a lot of reasons how cloning could benefit society. For example, medical purposes is a big one. The creation of transgenic animals could be drastically quickened with the use of clones. Also, the creation of stem cells could be revolutionized with the process of cloning.
Fun fact, transgenic animals are used for biomedical research and to understand gene function in the context of disease susceptibility.
Knowing that stem cells are mandatory to build, maintain, and repair our bodies throughout life, we can artificially clone them to do all sorts of impressive medical advances, like the possibility of growth for whole organs.
A few other reasons to advance cloning research is to make your favorite movie a reality. Jurassic Park shows a future where they are able to bring back extinct animals such as the dinosaurs.
This, in theory, could be possible. All you need is a well-preserved source of DNA from the dinosaur, and a closely related species that is currently living, and that could serve as an egg donor and surrogate mother. So realistically, maybe not.
Although cloning endangered animals is actually a much simpler process, so hopefully someday we will be able to save the giant panda. Other reasons to clone are to bring back your beloved pet and create breeding stock from slaughterhouse carcasses.
Now, one of the heavy questions surrounding cloning: should we advance this technology to be able to clone humans? Most scientists and lawmakers dub this idea immoral, but there are a few that support the idea of human cloning, claiming it is a solution to infertility problems and even ideas of cloning geniuses whose work could advance society.
Other far-fetched ideas is to fill farms with clones whose only purpose is to be harvested for organs. Despite these ideas, current research is focused on the creation of embryonic stem cells. And now some myths.
It is often thought that clones would magically appear as the same age as their original. Cloning is a way to create an embryo, not a robotic copy of a human, making this thought completely false.
Genes help determine traits, environmental influences, and also impact shaping an individual's physical appearance and personality. This shows that while clones carry identical genetics, this doesn't mean they will look identical to the original.
Cloning is not a new technology at all. Plant cloning can be as simple as cutting a branch from one tree and grafting it to another. And even artificial embryo twinning was first attempted over 100 years ago.
There are other ways to create clones. For example, asexual reproduction.
That is all for today's lesson on cloning with Dolly and the Sciencey Humans.
We hope you enjoyed and understood a little bit more about cloning and how it works, as well as some of its myths.
And remember, if we ever do create technology that could ease the cloning process, dinosaurs is not the way to go.